everyone, welcome back to Blood Talks. In today's video, we are diving into a detailed review of some key hematology test questions. We will be going over each question step by step, providing in-depth explanations, and helping you better understand the concept behind the answers. Whether you are studying for exam or just brushing up on your knowledge, this video will give you a solid foundation on some essential hematology topics. So, let's get started with the first questions. What does the potassium-sodium pump help maintain in red blood cells? The correct answer is B. The sodium-potassium pump is a vital membrane protein found in red blood cells and other cells. It maintains the balance of sodium and potassium ions across the cell membrane, which is crucial for cell function. Here how it works. The pump actively transports 3 sodium ions out of the cells and 2 potassium ions into the cells. This process requires ATP as energy for maintaining concentration gradients. By moving these ions against the concentration gradients, the pump helps keep intracellular sodium level low and potassium level high, which is essential for maintaining cell resting membrane potential. Cell volume regulations. The pump also plays a role in controlling osmotic balance, preventing cells from swollen and maintaining the shape of red blood cells. Hemoglobin F is a composed of 2 alpha globin chains and 2 The correct answer is D. Hemoglobin F is composed of 2 alpha globin chains and 2 gamma globin chains. Hemoglobin is a protein in red cells responsible for transporting oxygen from the lung to tissues and returning carbon dioxide to lungs. There are actually several types of hemoglobin, with hemoglobin A and hemoglobin F being the most notable. Hemoglobin A is made up of 2 alpha and 2 beta globin chains. It is the most common type of hemoglobin in adults, accounting for about 95 to 98% of total hemoglobin. Hemoglobin A has a moderate affinity for oxygen, allowing for effectively oxygen delivering to tissues. Hemoglobin F consists of 2 alpha and 2 gamma globin chains. This is the primary hemoglobin type found in fetus, which allows them to extract oxygen from maternal blood more effectively. Hemoglobin F has a higher affinity for oxygen than hemoglobin A, which is beneficial during fetal development. This higher affinity helps the fetus effectively draw oxygen from maternal circulations. Key differences. Hemoglobin F is predominantly in fetal and gradually decreases after birth, replacing by hemoglobin A. The production of hemoglobin F is controlled by the different genes than hemoglobin A, and its level can be influenced by certain medical conditions such as sickle cells and thalassemia. The differences in the composition led to the differences in the red blood cells property. Now that we know the property of hemoglobin A and hemoglobin F, what can we do about that? Or what tests would be benefit from the differences in their property? Or what tests can be beneficial due to the differences in their properties? That's correct, the KB test. The KB test is used to detect a quantity fetal hemoglobin in maternal blood, particularly in case of suspected fetal maternal hemorrhage. The unique property of hemoglobin F is the leverage that we can use because it is resistant to the acids. Let's look at it a little bit more for the KB test. 1. Acid resistance. In KB test, maternal blood is exposed to an acid environment. Normal adult hemoglobin is more susceptible to destruct under acid conditions, while hemoglobin F or the fetal red blood cells remain intact. This property allows the differentiations between maternal and fetal red blood cells. Staining. After the acid exposed, a stain is applied to color the remaining cells. The fetal cells containing hemoglobin F will retain their color, while the maternal cells containing hemoglobin A will lose their color because the cell membranes got destroyed. Quantifications. By counting the number of fetal cells versus maternal cells in a blood smear, CLS can determine the extent of fetal-maternal hemorrhaging, which is crucial in managing potential complications. And we can also determine how many vials of RHIG that we will be giving to the patient. Clinical importance of a KB test. The KB test is particularly important in case of trauma or certain conditions for the OB patients where there's a risk of bleeding from fetus into maternal circulation. By understanding the degree of fetal mother hemorrhaging helps guide clinical decisions regarding interventions such as the administrations of Rh immunoglobulins in Rh negative mother. Overall, the KB test is a valuable tool, allowing the assessment of fetal well-being and the management of potential complications for the mother. I have a video on how to calculate how many vials of RHIG to give, so please check that out at the end of this video. Where does the conjugations of bilirubin occur? The answer is C, liver. Let's take a look at bilirubin pathway from formations to exiting the body. Formations. Bilirubin is produced from the breakdown of hemoglobin in red blood cells. This process occurs primarily in liver and spleen, where hemoglobin is broken down into hemes and globin. The heme is further converted into biliverdin, which is further reduced to bilirubin. Transport to liver. Conjugated bilirubin, which is not water-soluble, binds to albumin in the bloodstreams and is transported to the liver. Conjugations. In the liver, unconjugated bilirubin is taken up by liver cells and converted into conjugated bilirubin or direct bilirubin. This process makes bilirubin water-soluble. Bile secretions. Conjugated bilirubin is secreted into bile, which is stored in gallbladder or directly released into small intestine. Intestinal process. In the intestines, conjugated bilirubin is converted by gut bacteria into urobilinogen. Some urobilinogen is reabsorbed into bloodstreams, while the rest remain in the GI tract, and that is what gives the stools its brown color. The urobilinogen that is not reabsorbed is excreted in the feces. A small portions of urobilinogens can be excreted through urine as urobilin, contributing to its yellow color. This entire pathway is essential for proper process and eliminations of bilirubin from the body. Preventing its accumulation which can cause jaundice, and here is a summary of the pathway. Which of the following is associated with glucose 6-phosphate dehydrogenase deficiency, or G6PD? The answer is B. The oxidized hemoglobin precipitate in the form of Heinz body. Oxidative stress may occur from infections, certain drugs, or during the immediate postnatal period. Let's talk a bit about G6PD. G6PD deficiency is a genetic disorder that affects the red cells. Individuals with this condition lack sufficient level of G6PD enzymes, which plays a critical role in protecting red cells from damaging caused by oxidative stress. When exposed to certain triggers such as infections, certain medications, or certain foods like fava beans, people with G6PD deficiency can experience hemolyzing, where red blood cells break down faster than they can be produced. This can lead to anemia, fatigue, jaundice, and other symptoms. G6PD deficiency is inherited by X-linked recessive manner, meaning it's primarily affecting males, although females can also be carrier and may experiencing symptoms. Diagnosing typically involve a blood test to measure MSI level, and a hemoglobin test may show evidence of hemolyzing. To manage people with this is to avoid triggers, and in severe cases, treatments, hydrations, blood transfusions, or other interventions are needed. Conclusions: G6PD is a vital enzyme for red blood cell health, and its deficiency can lead to significant clinical challenges. Awareness of the conditions is important, especially in population at risk, to prevent complications and managing the condition effectively. That's all I have for today. Did I miss anything? If you have any questions, please let me know. I'm more than happy to answer them. If I don't know, I will try my best to find out for you. Also, keep in mind that the information I put together here is the general practice at the moment. As time changes, certain practice may change, and different institutions may have different policies. So please keep an eye out for that. If you like my video and think it's helpful in any way, please share it with your friends, and I shall see you all next time. As always, remember, your blood tells you the story of your health. Thanks for watching. Bye!